I am going to continue the stem cell theme, but talk about a new pathway that we discovered, which we believe is very central to cancer biology, to a very significant percentage of human cancers. But it was discovered through a window on embryonic and pluripotent stem cells. So what I'll talk about is the stem cell regulator Lin28 and its role in human malignancy. My laboratory for many years has been interested in the intersection, the sort of common themes of stem cell biology and cancer biology, and that has come into very direct focus with our study of the Lin28 LET7 pathway. But in stem cells, we've been focused on the notion of somatic cell reprogramming, the process by which you take adult somatic cells, introduce genes that are typically carried in embryonic stem cells, and revert the somatic differentiated cells back to their embryonic potential. And essentially all of those genes, the transcription factors that drive reprogramming, OCT4, SOX2, MYC, and others, are in other contexts transforming genes that drive oncogenesis. So clearly the same pathways that are involved in resetting the differentiated state of a cell are involved in resetting the cancer cell phenotype. And for many years, there's been the notion that cancers in some contexts involve a de-differentiation process. And I think our insights into the reprogramming process are going to teach us about cancer. And as you see, these pathways are common. So how did we, after my lab for many years was interested in the leukemia oncoprotein BCR-ABL, we then morphed into a stem cell focus because of our interest in the effect of BCR-ABL on the hematopoietic stem cell and the use of bone marrow transplantation in treating chronic myeloid leukemia. But we ended up then coming back to cancer, and I want to tell you that story because I think it's actually quite interesting. So we were interested in just a very basic problem, and that is trying to understand the first fate choice in the mammalian embryo. So when a egg and sperm come together, the zygote is 2 cell, and then it divides into 4 cells, and then 8. And at the 8-cell stage, it's essentially equivalent. Each of those cells are equivalent. And then this remarkable division occurs, a segregation of the inner cells, which adopt a fate that they will become the cells of the embryo proper. And they do that in the process of morular compaction to form the inner cell mass. And a number of years ago in the mouse, and then in 1998 in human, these cells were cultured in vitro as embryonic stem cells. Now, at the same time that these inner cell mass are being set aside, there is the segregation of the external rind of cells of the developing embryo, the so-called trophectoderm. These occur under the influence of different transcription factors, but they set up their own stem cell pool, and that can be captured in a petri dish as trophectodermal stem cells. And this is really the first fundamental segregation of tissues in the developing mammal. So we know from really decades of manipulation of mouse embryonic stem cells that they are restricted. They do not contribute to the placental or trophectodermal or extraembryonic tissues. But we came across a very surprising result. We were interested in asking whether we could convert the embryonic stem cells to a more malignant phenotype. Embryonic stem cells are known for their ability to form what are rather benign encapsulated teratomas. In fact, we use teratoma formation as an assay for defining the pluripotency, the ability of embryonic stem cells to give rise to many tissues. But they don't, they aren't traditionally believed to give rise to trophectoderm. But we added the RAS oncoprotein into embryonic stem cells. We induced it in a conditional way. And what we observe is really quite striking. was that we could convert teratomas now into a very aggressive malignant and fatal tumor in engrafted mice. And when we looked at that tumor, we were quite surprised to diagnose it as a choriocarcinoma, as a tumor, a malignant tumor of extraembryonic trophectoderm. So this suggested to us that fundamentally, by activating and toggling the RAS pathway, we had fundamentally reprogrammed the embryonic cell to an extraembryonic fate. And so that raised then, in our minds, a very interesting question of how was RAS being regulated in the early embryo? And Janet Rossent and others had actually demonstrated that this dichotomy was in fact in part regulated by signaling through the FGF receptor via the RAS MAP kinase pathway. But we then started to look more broadly at regulators, and we took note of the fact that it had been shown by Frank Slack and colleagues that a prominent regulator of RAS, in particular in the early embryo, as had been shown by Scott Hammond's group, was a family of microRNAs called LET7. So we then wanted to dig deeper into how this family of microRNAs was regulating the RAS process. And what we learned from Scott Hammond and then reproduced, and this is the work of Srini Viswanathan, a very outstanding graduate student in my lab at the time, was that this family of microRNAs was expressed in the early embryo as a primary transcript, but it wasn't processed. So it suggested there was some mode of regulation. And in fact, what Srini went on to do, working with Richard Gregory, who's a junior colleague who was a maven of microRNA biochemistry, biochemical processing, Srini could show that if you added extracts of embryonic stem cells to an in vitro microRNA processing assay, you got the same blockade as you saw in embryonic development. So here you can see this is just showing the primary transcript across embryonic stem cell differentiation or in the early embryo, but only in the mid-gestation or in later stages of in vitro differentiation do you actually see processing. So this suggested there had to be an inhibitor of this microRNA. And indeed, working with Richard, we were able to use a pull-down and mass spec approach, and we pulled out a protein. And that protein was called Lin28. Now, we had to go back into the literature to figure out what Lin28 was. Lin28 had originally been described by Bob Horvitz and Victor Ambrus when they were doing very basic screens for genes that would disrupt the developmental timing of worms. And this is a very important example of where just investigations in fundamental pathways of biology will sometimes yield insights that are unanticipated but have really profound impact on human disease. And this was clearly an example. So Lin28, we now know, and I'm just going to summarize, is a critical element of the regulation of processing of the LET7 family of microRNAs. Now, microRNAs are processed in the following way. They're transcribed in the nucleus, and then they are exported into the cytoplasm and processed into a terminally differentiated microRNA, which then interacts with messenger RNAs and regulates in a post-transcriptional way the expression of genes. And this LET7 family was particularly interesting because it had been implicated as a tumor suppressor family by virtue of the fact that it had a negative regulatory effect on the expression of oncoproteins like RAS and MYC, but other components of the cell cycle regulatory machinery like the cyclins. Now what Lin28 does is it binds to the precursor form of LET7, and then it assembles an enzymatic complex around the microRNA, which leads to the derivatization by a poly-U tail that targets it for destruction. So in the presence of Lin28 protein, you have no fully terminally differentiated LET7 microRNA, and what this does is it de-represses all of the LET7 targets. Now, what we also know is that LIN28 is a protein that not only interacts with LIN28, with LET7, but it also interacts directly with messenger RNA. And let me just show that it will interact with the messenger RNA where it also binds and influences translation. So there are LET7 dependent and independent effects of this RNA binding protein. So what would we predict? If this is the maturation of the LET7 family of microRNAs acting across these targets, we might anticipate that an inhibitor of the inhibitor would release these targets and should be oncogenic on its own. And so our prediction was LIN28, and I should point out LIN28 exists in two paralogs, so there's an A and a B. But we predicted that it would be involved in cancer. So one of the first things we did was examine the expression of LIN28 in the NCI-60 panel of cancer cell lines. Now, LIN28, I should additionally mention, is an embryonic regulator. It's not just in embryonic stem cells, but it's highly expressed in a broad range of tissues in the early embryo. And then about mid-gestation, it's basically silenced virtually everywhere. So this expression pattern, seen in 8 or 9 of the NCI-60, really reflects the reactivation of what has emerged as an oncofetal program in the context of these cancers. Now, we took note of the fact that one of the cell lines in which it was most highly expressed was the blast crisis chronic myeloid leukemia cell line K562, which was an old friend of ours. And we were able to then show that additional CML blast crisis cell lines were also overexpressing LIN28. So we then asked whether LIN28 upregulation might play a role in the ongoing proliferation of K562 lines. And indeed, we could use a short hairpin to knock down LIN28, in this case LIN28B. We could show that the cell lines would reduce their proliferation rate and, interestingly, undergo a very dramatic degree of cell differentiation. So LIN28 in this context appeared to be blocking the differentiation of the CML cell lines. We then went back to a model that I had done as a graduate student in which we can introduce the BCR-ABL oncoprotein into the bone marrow of mice, and they will go on to develop a CML-like leukemia. One can then actually show the oncogenic cooperation of other genes implicated in acute myeloid leukemia, as has been shown by Gary Gilliland, like the NUP98 HOXA9 fusion protein. When you introduce these two genes together, you convert the pathology in the mouse from a chronic leukemia to an acute blast crisis. And indeed, we wanted to know whether LIN28 would do the same. And so, a complicated slide, but I would just point out that in the primary transplanted animals, this is the classic latency of the chronic phase of the disease, which is primarily myeloid, but in the context of adding either the NUP98 HOXA9 or LIN28 on top of BCR-ABL, one converted this model to a blast crisis acute-like phenotype. Then we were able to show, in collaboration with a number of folks who provided clinical samples, Jerry Radich, Charles Mulligan, Tim Hughes, and others, that there did seem to be, in paired samples of chronic, accelerated, and blast crisis, a tendency for upregulation of this LIN28 pathway. So then we asked more broadly whether LIN28 was a common mechanism of activation across a diversity of human cancers. And so turning to tissue microarrays, essentially, tumor microarrays, we were able to demonstrate in a very large series. This is just measuring LIN28A, and as it turns out, LIN28B is in fact even more frequently upregulated, but I'll show you the data. We saw positivity in colonic adenocarcinoma, breast adenocarcinoma, lung squamous, and really a a very large range of tumors. Now, in particular, we noted that there was a tendency for LIN28A to be upregulated in pediatric tumors, and in particular, a majority of hepatoblastoma, a significant number, about 30% of Wilms, and every germ cell tumor. Whereas LIN28B tends to be upregulated more in the adult range of tumors. So just to talk a bit about the role in germ cell tumors, because we've published this, it turns out LIN28 plays a central role in the regulation and maturation not only of the early embryo, but of the germ lineage itself. And so overexpression of LIN28 in developing mice actually causes an expansion of primordial germ cell pools. And as we first noted, just by expression analysis, the LIN28A and B paralogs are upregulated and overexpressed in a broad array of germ cell tumors, and indeed it is even a marker that's more prevalent than OCT4, which has emerged as a dominant marker used by pathologists. And indeed there are 4 different groups of human pathologists who have documented the application of LIN28B to the diagnostics of germ cell tumor, and it's essentially now part of the normal practice of typing these tissues. So let me speak in detail about one model that we've set up, and it's a model which once again teaches us that serendipity can often be a more interesting happenstance in science than actually finding something you're setting out to do. So this is the work of Ahia Urbach, who's an Israeli postdoc who's just about to return to Israel. And he, noting the high correlation between LIN28 and germ cell tumors, wanted to set up a mouse germ cell tumor model, because really they don't exist. So he engineered strains of mice to express the LIN28A oncogene when a Cre recombinase would be expressed so as to remove the stop codon ahead of this cassette. He chose to drive Cre off of a germ lineage-specific promoter called VASA, which should, in practice, generate tissue-specific overexpression in the germ lineage. Well, it turns out that VASA is also expressed in the human egg, and so what you end up getting is a ubiquitous constitutional deletion. And yet, even with LIN28 activation in all the tissues, there was a predominance of kidney lesions. And in fact, we saw significant, even massive kidney tumors. And when we looked at the histology of the tumor, we noted that LIN28 had been overexpressed. And by microarray, looking at the genes that were enriched in response to LIN28, what we found was a signature, both histologically and genetically, of the pediatric cancer Wilms tumor. Now, Wilms tumor is really a fascinating disease. Dan Haber at the Mass General Cancer Center has done work on the WT1, which is a loss of function mutation that is involved in about a third of Wilms tumors. What happens in normal kidney development is this remarkable process of mesenchymal induction, where the developing ureteric bud invades into the surrounding mesenchyme and causes a mesenchymal to epithelial transition such that you get the aggregation and formation of the developing collecting system that ultimately forms this remarkable structure, which is the nephron, which is the functional unit of the kidney. Now, I never imagined that I would find kidney development so fascinating, but by coming into the Wilms tumor area, I've now actually become focused in this. So what it turns out is that this period of nephrogenesis in the developing kidney is pretty much only throughout embryonic development, and then very soon after birth, about day 2 in the mouse and by the first couple of weeks in the human, every single nephron in the developing kidney is in place. So when we're an adult, if we suffer any kind of significant kidney injury, nephrotoxic kidney injury, we don't regenerate that. We end up with kidney failure. So what's really fascinating about Wilms tumor is it's not just the most common pediatric renal cancer, but it really is a disruption of this process in such a way that there's a prolongation of the nephrogenic zone. So what we see in the mice kidneys that have overexpressed LIN28 is we see this remarkable expansion of the nephrogenic zone, but we see ongoing differentiation. So the animals end up with essentially an increased frequency of nephrons, some of which, because of ongoing proliferation, deteriorate into actual tumors. So we went back and said, well, is this the same histology that's happening in the human? And indeed, we found that working with Kathy Pritchard-Jones in the UK, who has a very, very large database of human Wilms, that over 30% of human Wilms have activated this LIN28 pathway. And we can show by immunohistochemistry that, in fact, what is happening in the mouse tumors is that we are prolonging the period in which this cap mesenchyme, which is kind of the primordial stem cell of the developing nephron, this persists in the tumor. So, of course, in the context of overexpression of LIN28, one gets a downregulation of LET7. So we went back in normal kidney development and we asked, is there a normal role of LIN28 in the kidney? And indeed there is. So when the early cap mesenchyme is proliferating, LIN28 is highly regulated, upregulated, and then it's silenced as you get this conversion to terminal differentiation. Indeed, here's the marker of cap mesenchyme, a factor called 6-2. In the normal kidney, LIN28 marks the same population of cells and then is silenced. And so with transgenic overexpression or oncogenic reactivation, we expect that this is then continued. So we then went to see if we could dissect the particular cell of origin of the Wilms tumor. We were predicting that it would be this cap mesenchyme cells. But interestingly, when we expressed LIN28 in each of these specific subtypes, which map either cap mesenchyme, stroma, or the ureteric bud, we didn't get a tumor. We only got a tumor when we expressed in the WT1 positive population, which is the precursor of all of these different lineages. And we're still trying to understand that and trying to reconcile how that dysregulation relates to the known expression of LIN28 in normal development in the cap mesenchyme. But WT1-CRE is now our animal model, which gives us a very predictable incidence of Wilms tumor formation. So what's our mechanism? Well, in the natural process of kidney development, you have, in the early stages of embryonic development, a primarily self-renewing and expanding cap mesenchyme population, which converts to differentiation by postnatal day 2, or the first week or two in humans, postnatal in day 2 in mice, so that only mature nephrons develop. That's normal development. Vicky Huff's group has modeled the WT1 ablation, which is the mechanism of Wilms tumor that Dan Haber and others had demonstrated. And here what you get is essentially unextinguished self-renewal of this early progenitor population, and that characterizes probably a third of human Wilms tumor. And now we have added a second model, which entails a slightly different histology, which we understand from the renal pathologist is more characteristic of the diversity of Wilms tumors that are seen, because you see not just an arrest in differentiation of Wilms tumor, but you see a tendency for these differentiating nephrogenic arrests as well as some degree of blastemal formation. So you see sort of all of these critical elements. And this, what we believe, is because LIN28 is not an abrupt break on differentiation, but rather simply hearkening back to its original role in the worm it's prolonging the periods of self-renewal and delaying the developmental timing of this tissue. So, can we prove that LIN28 is essential for maintaining the tumors? We show it's involved in initiation. But because we can regulate the expression of this oncoprotein under the control of doxycycline, we can induce the tumors with exposure to doxycycline, and then we can withdraw the doxycycline and see whether or not the tumor has been sustained in the absence of LIN28. And what you can see here, just by total kidney size, is it is not. So in the absence of LIN28, the tumor will then regress. Furthermore, we can then ask whether or not its effects on the tumor suppressor microRNA family, the LET7 family, is the principal mechanism by which it's deregulating these tissues. And what we do is we breed into the LIN28 transgenic mouse, a mouse that is upregulating a form of the LET7 microRNA which is resistant to the activities of LIN28. And that's a complex chimera of the microRNA that can't bind LIN28. And in this experiment, what we can show is that here's expression of the LIN28 transgene itself, making a large kidney destined to be a tumor. But in the presence of the LET7, the LIN28 resistant form of LET7, we see no tumor formation. So in this, we conclude that the LET7 is dominant over the LIN28, and we in fact believe that Wilms tumor is really a disease of the deficiency of LET7. Now, what's really fascinating, and will probably take us forward, is the idea that we can toggle the dose of LIN28 in such a way that we can actually grow a larger kidney with a higher density of nephrons. And this is just shown here. In those experiments where we withdraw the LIN28 and we don't see tumor formation, what we do see is a commitment to the differentiation of a larger density of nephrons. Now, we're currently testing whether these kidneys are, in a sense, super filterers. We're trying to determine whether we've actually enhanced the filtration function of these kidneys. Because what we would like to envision is to be able to reactivate nephron development in the context of an injured adult kidney. Because if we could reactivate the ability of the adult kidney to form new nephrons, we would have a potentially transformative new strategy for treating kidney failure. And we have reason to believe, based on preliminary evidence, that in fact we're going to be able to achieve this. Now, in the cancer realm, I haven't talked about some of the other models, but as has been published by Anil Rusti's group, terribly scooping us because we have the same data, LIN28B, driven in the colonic mucosa, forms a very remarkably invasive adenocarcinoma of the colon. So LIN28, we've shown now, is overexpressed in about 50% of human colon cancer, and we've modeled that very, very effectively in mice. John Maris's group, as well as European groups, have demonstrated that LIN28 is a central driver of neuroblastoma, and we have a paper under review at Cancer Cell that reports its role, as we detected in the original studies, in liver cancer. So can we drug this? And that's the last word I'll leave you with, is that my colleague Peter Slees, who's a structural biologist, has solved the co-crystal structure of the LIN28B protein with its LET7 microRNA target. And indeed, what we see here is a prominent groove that through mutational studies we know is dictating the specificity of this association. So the LIN28 protein RNA interaction involves a very potentially druggable target. Now, this would in fact be a novel class to drug. It would be a drugging of an RNA protein interaction, and I think there's still questions about whether we could achieve the kind of potency and selectivity that we would need to be able to to deliver a drug. But I should say that Peter, his lab, has configured fluorescence polarization screens, and we've been working with him on cell-based assays. And in the first flurry of about 150,000 compounds done at the ICCB Screening Center at Harvard, there's been a very, very encouraging group of relatively high potency hits, which have been refined through secondary assays that involve disruption of the protein complex that's formed around LIN28 and LET7. And we're currently documenting whether these will have activity in cell-based assays. But our hope is that this will be a druggable target that we'll be able to take into future clinical translation. So let me summarize. The original work was done and published in 2008, but it really has essentially dusted off this old protein, the LIN28 protein, that had originally been described by Ambrose and Horvitz in 1984. And the LIN28 is a known regulator of LET7, and LET7 is a tumor suppressor class of microRNAs, and so this really puts us very squarely in the setting of a cancer model. I didn't have time to speak about it, but this pathway it's a fascinating pathway that we've studied in a number of physiologic contexts. Genetic variation and GWAS studies of human height and the timing of human puberty have found variations in this pathway, and we've actually been able to demonstrate not only effects of organismal growth and puberty in mice, but we've been through studies of the mechanisms there, we've actually implicated this pathway in glucose control and diabetes. And in fact, we do believe that one of the effects of LIN28 LET7 on the cancer cell is to stimulate the Warburg-like high glycolytic flux metabolism of the tumor cell. Just to mention, coming out online in November is a fascinating observation that once again we can reactivate this pathway in adult tissues and confer on adult tissues the heightened tissue repair and regeneration response that we recognize in juvenile organisms. And so once again, what we're doing is really toggling the developmental age of these tissues so that they react and repair like an embryonic tissue. But in cancer, I gave you the story about it being a driver of Wilms tumor and mentioned just briefly its role in these other tumors. And I mentioned once again briefly, but we'll continue to explore the fundamental molecular targets of the LIN28 LET7 pathway. We know that LET7, and we've reported its importance in regulating a number of targets in the PI3 kinase mTOR pathway, but what we're really focused on now are the non-LET7 dependent effects on the metabolic pathways and the Warburg effect. This pathway is an embryonic pathway, and I think it's an example of where just fundamental studies into embryonic stem cells, the biology of human embryos, has taught us about something which now is really, I think, of central importance to cancer. Another good plug for investments in basic research, because this pathway, which is an oncofetal pathway, is co-opted in tumors, and we do believe it's going to be druggable. So the work is now about 50% of my lab. I want to point out the contributions of Hao Zhu, who is a really outstanding oncologist. He trained in the Harvard program. I wish we had been able to keep him here, but he's now an independent scientist at UT Southwestern. He had a very good colleague in Xichang Ing, who is a graduate student who just defended last week in my lab and is off to become a fellow at the Genome Institute in Singapore. Achia Urbach, a postdoc working on the Wilms platform together with a graduate student, Elena, and Achia is about to return back to Israel. The early studies that we did on the biochemistry of LIN28 has been done with Richard Gregory, and he's really done remarkably beautiful work to elucidate the other critical control elements of the biochemistry of LET7 processing. John Powers works on neuroblastoma. We have several other folks who I mentioned is also working on this pathway. Daisy Robinson on the liver, Kaloyan on signaling, and I think this is going to turn out to be just a fascinating target, and I hope we actually end up getting it blocked and moved into the clinic. So thank you very much for your attention, and I'm happy to take questions.
Not all treatments for Wilms tumor are effective. Is there a difference in response to whether or not the tumor is WT1 generated or the LIN28?
Yeah, it's a very good question. So there are a number of different subtypes of tumor, and we are actually working with Catherine Pritchard-Jones on this issue. She has a tremendous database. She has them molecularly subtyped. We now have the immunohistochemistry on the LIN28. There seems to be mutual exclusivity, so that if you're WT1 positive, you're not LIN28 positive, and vice versa. If you're LIN28 positive, you're not WT1 deleted. So we are just in the midst of doing those clinical correlations. We've assembled a few of the Wilms tumor cell lines, 3 of them. Those seem to be LIN28 expressors and not defective in WT1. And we're working on studies of demonstrating the sufficiency and the necessity of LIN28 as a transforming gene there. Bruce.
George, I don't want you to disclose any confidential information, but what is the strategy for developing an inhibitor? Is it focusing on oligonucleotide fragments, or is it small molecules?
Yeah, so other groups have actually shown that you can develop small oligonucleotides that mimic the sequence of LIN28, and in fact they will displace the LIN28 LET7 interaction. So you could take an aptamer-like approach. Of course, then the issue becomes delivery and cell penetration. So we've avoided that, and we've simply taken a classical small molecule approach. And at least in the biochemical assays, we have very good chemical matter.
Are the compounds in some way modeling the surface of the oligo or the LET7?
We don't, no, it doesn't look like that, at least on the face of it. We don't really know how they're acting, but Peter's group, given that they're a structural group, is actually trying to soak some of these molecules into the co-crystal and also crystallize them de novo. Great, thanks.